Hello all, introducing the topic crossing over. Previous session we have learned what linkage is. Linkage is the genes which lie very close to each other will be inherited generation after generation because they lie very close to each other. Now next is crossing over. Crossing over is the exchange of the DNA or the genetic material between the non-sister chromatids, the random exchange of DNA between the non-sister chromatids. These two are the sister chromatids. This same purple colored one are the sister chromatids. These two are the non-sister chromatids. Non-sister chromatids. So the exchange of DNA between the two non-sister chromatids of homologous chromosome is called crossing over. And because of this crossing over, actually the genetic variation happens because of the crossing over or because of the exchange between the exchange of the DNA or the genetic material. Variation happens in inheritance, while inheritance. That's why the offsprings or the does not look as same as the parent is. So when crossing over occur, you can see here, see these two chromosomes will undergo crossing over. These are the non-recombinant chromosomes, the purple ones, the top layer. These two will not undergo any recombination, and these two which lie very close to each other, non-sister chromatids which lie very close to each other undergoes crossing over, and that result in recombination which result in the variation of the chromosomes. So crossing over is defined as the mutual exchange of segments between the non-sister chromatids of the homologous chromosome resulting in the recombination of genes. That is the definition. Once more I'll say crossing over is the mutual exchange of the segment between or the genetic material between the non-sister chromatids of the homologous chromosomes resulting in the recombination of genes. And usually this recombination takes place in the gametes. And the crossing over and independent assortment are the major mechanisms that produce new combinations of genes. That's what I said. As a result of this crossing over, there is an exchange of genetic material, and as a result, new variants or the offsprings with showing changes from the parental types are produced. So, the natural selection can act upon these new combinations to produce organisms with maximum fitness. As a result of recombination, the new combination of genes will be formed. Then the natural selection will act on that individual or that genes, and only those viable or efficient recombinations will be able to survive. That's known as natural selection. Now, this is, you can see the chromosome A and B. These are the two chromosomes. This is shown by capital letter and this is shown by small letter to differentiate. So, chromosome A and chromosome B. And this is undergoing crossing over. Crossing over means the non-sister chromatids which lie very close to each other undergoes crossing over. And like this, crossing over happens like this. See, this chromosome is exchanged with this arm of the chromosome. So small letter E, small letter F, small letter G is now changed to small letter E, small letter F, small letter G, and capital letter H and capital letter I. That means this part has underwent the change or exchange, and as a result the chromosomes which are formed are called recombinant chromosomes. So those non-homologous chromosomes which lie very close to each other will undergo crossover like this and crossover, and they will, after that what happens, they will separate like this. They will move to opposite poles. As a result, new combination is formed. And this picture clearly explains how this exchange has happened. See, this much part has now joined with these small letter H has joined with the capital letter sequence of this chromosome B, and chromosome A has underwent achieved a session part of capital letter H and capital letter I, and so new combination is formed as a result of recombination. That is crossing over. This is what is a simple mechanism, just pictorial representation of what is happening during the chromosome crossing over. So the important thing is two non-homologous chromosomes or the non-sister chromatids should lie very close to each other. So the prevalence, see, the prevalence of recombination is dependent on the distance between the linked genes. So the occurrence or the chance of crossing over between the chromosome depends on the distance between the linked genes. If the genes are very far to each other, the chance of crossover is very high. That is opposite to that of a linkage. See, this part, this is a chromosome A, B, C, and here small letter A, B, C. And this much part is undergoing a crossing over like this. So what happens? Those parts which lie very far away are having more prevalence to crossing over. The prevalence of crossing over or recombination is more when the genes are far apart. Then, based on the types of which the crossing over, the crossing over is affected by lot many things. That is the sex of the individual. That is the tendency of reduction of crossover in male mammals. Male mammals, the tendency of crossover is less. Then mutation will reduces the crossing over. The factors which affect crossover are 1. Sex, 2. Mutation, 3. Temperature, 4. X-ray effect, 5. Age. That is the males have less chance of crossing over, and mutation reduces the crossing over. High and low temperature variation increases the percentage of crossing over. That means for a crossing over to take place in a cell, there should be an optimum temperature. Below or above that temperature, the crossing over will not be that much efficient. Next, X-ray effect or X-ray irradiation will increase the crossover near the centromere. And about the centromere, the one thing which we have to be keep in mind is that near the centromere, the crossing over chance is very less. Near the centromere, the chance of crossing over is very less. Only the part of the chromosome far from the centromere shows more crossing over. So, X-ray, as a result of the X-ray effect or X-ray irradiations will increase. This normal phenomena or X-ray irradiation will increase the crossover near the centromere. So, usually the crossing over is less near the centromere. Next is age. Older age increases the rate of crossing over. As the individual is getting older, the rate of crossing over is more. Now, the significance of crossing over. What is the significance of crossing over? It produces, of course, the first and the important thing is it produces new combination of traits or new varieties. The parental, that is phenotypically, if you look, the offsprings will not look exact, are not exact copies of their parents, and that is because of this phenomenon of crossing over. As a result of crossing over, new genetic combinations are formed and new traits are formed, and that is what is promoted by nature too. Nature don't want a monoculture or individuals of same type. It prefers, or nature look for varieties. So this first significance of crossing over is it produces new combination of traits. Then 2. Through crossing over, the segments of homologous chromosomes are interchanged. That is what we have learned. That is interchanged. 2 chromosomes will lie together and they will undergo interchange. And as a result, new characters or new genetic variations are produced. New characters or new genetic variations will be produced as a result of crossing over. Then, crossing over plays a very important role in the field of breeding. Crossing over helps, and I said new varieties means new combinations of chromosomes result in producing new varieties, like in plant breeding or animal breeding. In experiments, what usually is done is the wild type variety will be crossed with the yielding varieties, the plants or the animals. So such combinations are obtained as a result of crossing over. So the significance of crossing over, important, it's new combinations are formed, two new characters or genetic variations are formed. Then crossing over have a very important or a significant role in the breeding experiments of plants as well as animals where in combining the wild characters with the yielding varieties like that. Now, the different types of, next is the types of crossing over. The crossing over is of two different, there are different, based on different categories, there are different types of crossing over. First one is germinal crossing over or meiotic crossing over, and two, somatic or mitotic crossing over. Germinal crossing over. As the name signifies, the crossing over which occurs in the reproductive cells or germ cells is called germinal crossing over. And in gametes, the division which is happening is meiosis. So it is also called meiotic crossing over. So germinal crossing over takes place in the gametes during meiosis. So it is called germinal or meiotic crossing over. Crossing over which occurs in the reproductive cells in the prophase. In meiosis, it is happening in the prophase. That is important. So only this type of crossing over has the genetic significance. Actually, the crossing over in germinal cells is more important, because only if recombination takes place in the germinal cells, that exchange or new traits will be produced. So that is very, very significant. So meiotic crossing over occurs in most sexually reproducing organisms, or only very few exemptions are there among the insects. So that is germinal or meiotic crossing over. It takes place in gametes during the prophase of meiosis. Then 2. Somatic crossing over or mitotic crossing over. Why? In mitotic cells, in somatic cells only mitotic division or multiplication division takes place. There is no reduction in the chromosome number in somatic cells. So the somatic or mitotic crossing over is actually taking place in the non-reproductive cells, and it is very rare compared to the germinal crossing over. The somatic crossing over is very rare. It's very rare. And the effect of the mitotic recombination of alleles is not transmitted to the next generation. So there is no significance in the somatic crossing over. Why? Because it's not inherited. That if any crossing over take place during mitosis, that will be remaining in that particular organism only, and that will be ended at the by the death of that organism. That is not at all inherited. So crossing over which is occurring in the germ cells is of significance, or it is more important. See, these are the sister chromatids. The blue color chromosome and the red color chromosome shown here are the sister chromatids. These terms you have to keep in mind. So later on we will be talking only about the bivalent, the crossover, the chromatids, the sister chromatids, like that. So to understand that terms, I'm just showing this picture. See, this blue color chromosome and the red color chromosomes, they are called the sister chromatids. The blue and the red, they are separately, this is a sister chromatid, these are sister chromatids. Then blue and red are called non-sister chromatids. Blue is sister chromatids, red sister chromatids. If you draw like this, it is non-sister chromatids. Now, see, on this chromatids, you can see the genes. So the genes are present on this chromosome. This capital letter R, capital letter G, small letter R, small letter G, etc. are the location of genes on the chromosome. So the location or positions where the genes are found on the chromosomes are called loci. See, as a result of crossing over, see, look at the next picture. As a result of crossing over between the non-sister chromatids, see, these two are called the non-sister chromatids. These two are the non-sister chromatids. Blue separate, red separate. രണ്ട് blue ചേർന്നിരിക്കുന്നത് sister chromatids. രണ്ട് red ചേർന്നിരിക്കുന്നത് sister chromatids. ഒരു ബ്ലൂവും ഒരു റെഡും കൂടി തമ്മിൽ ചേരുമ്പോൾ അല്ലെങ്കിൽ റീകമ്പൈൻ ചെയ്യുമ്പോഴേക്കും we say they are non-sister chromatids. Hope that is clear. So the crossing over takes place between the non-sister chromatids. See, a red chromosome, it's undergoing recombination here at this part. So the capital letter R is undergoing recombination. As a result, what happens? Here there is a exchange, or here there is a change in the chromosome combination. Here you can see a small letter G. Look at this position. You can see a small letter G. Actually it was capital letter G. So there is a black segment have come here, and here there is another black segment. So there was an exchange of genetic material, and this is called recombination. And this result in the form, and as a result the chromosomes, it will separate, and these are called the daughter chromosomes. And those particular positions where the crossing over is taking place is called chiasma. That is important. You might have learned this in the meiosis also. Chiasma. The points at which the chromosome cross over is called, the point at which the chromosome cross over is called a chiasma. See, this black color one and the white color one, it undergoes a crossover, and it is taking a imaginary shape like that of a X, and this point is called chiasma. See, after this recombination, this black chromosome got a white part of the other one, and the white one got a black part of the other one. That means what? The exchange of genes has happened between the two non-sister chromatids. So that is the type. Then during gamete formation, what happens is, during gamete formation, what happens is that the intact chromosome, they lie together side by side, and they undergo recombination. And this chromosome sets which lie together, this is called the tetrad, and that is the chromosomes which lie together, the 4 chromosomes in this is called a tetrad. This set is called, the chromosome which lie together is called a tetrad. Thus 2 of the 4 chromatids are affected during the crossing over at a locus. See, so how many chromosomes are affected? Only 2 chromosomes. Why? The 2 chromosomes are here. This chromosome is not at all affected. It has the same sequence. And here also, this chromosome is not at all affected. No recombination. But the chromosome which is affected is one blue of this side and one of this side. So in total, what happens? Only 2 of the 4 chromatids are affected in crossing over, because they only form a chiasmata. Only a chiasmata is formed between the 2 chromosomes. So only 2 chromosomes is having some structural difference, or there is genetic difference only in 2 chromosomes. In which chromosome? Those which lie very close to each other, and on which a chiasma is formed. So only 2 chromosomes, this number is important, only 2 chromosomes of the 2 chromatids, 2 of the 4 chromatids are affected in the crossing over at a locus. Now let's see the mechanism of crossing over. The chromosomal crossing over occurs during the prophase of meiotic division 1. It is taking place in the prophase of the meiotic division 1, in which the segments of the homologous chromosomes are exchanged. And the events which is happening in the homologous chromosome in the crossing over are: one is synapsis. Synapsis is the pairing of the homologous chromosomes and the formation of a synaptonemal complex. So during zygotine stage, during zygotine stage of the meiosis, the homologous chromosome will align side by side. See, this is what is happening during the chromosomes will align side by side during the zygotine stage, and this pairing is called synapsis. Synapsis means the aligning or the pairing of the chromosomes during zygotine is called synapsis. So during synapsis, the chromosome will lie parallelly or aligned side by side, and this is called a bivalent. This structure is called a bivalent. This is called a bivalent. Bivalent means the 2 chromosomes, the 2 homologous chromosomes aligned side by side, and that's called a bivalent. And this pairing is called synapsis. And what is happening during the synapsis? You can see a ladder-like structure. See, between the chromosomes, between the non-homologous chromosome, you can see a blue color line, and that is a ladder-like structure is formed between the non-homologous chromosome. What is this structure called? This is called the synaptonemal complex. It is actually a complex having proteins and all. We will discuss the detailed structure of synaptonemal complex in cell cycle. So, during zygotine stage, the non-homologous chromosomes will lie side by side, and this pairing is called synapsis. And these chromosomes which lie, then this stage is called, this is called a bivalent, and a complex is formed between these 2 chromosomes, and that's known as a synaptonemal complex. Synaptonemal complex means DNA plus proteins. Then it consists of, and this synaptonemal complex consists of a 3 parallel axis. Actually, the synaptonemal complex is like this. It has a 3 parallel axis like this, which is composed of proteins, and the central axis is connected to the lateral axis by cross fibers. Some fibers will arise from this axis. Much details you can learn in the cell cycle. Thus, the synaptonemal complex will hold the homologous chromosome together, and such a complex formed by the pairing of a synapsed chromosome is called bivalent. Bi means 2. So, 2 set of chromosomes will lie together, and so a bivalent is formed. That is the first step in crossing over. So, first step in crossing over is the chromosome have to lie side by side. They have to come closer, and that is known as synapsis. And synapsis is happening because of the presence of a complex called synaptonemal complex. Then, second step. First synapsis is over, the chromosome has come together, and a complex of fiber-like mesh is formed between the chromosome. The next step is duplication of chromosomes. That is, the homologous chromosome will undergo duplication. The homologous chromosomes will undergo duplication, and as a result, it forms a tetrad. As a result, it forms a tetrad, means 4. So, during packaging, and this takes place during packaging. This takes place during packaging. Packaging is a stage of meiosis. So, duplication of chromosome is the second step, and duplication takes place in packaging. And during packaging, the bivalents undergo duplication and form a structure called tetrad. And the homologs are still held in position by the synaptonemal complex. Then, next is, third step is crossing over. Synapsis, duplication, crossing over. During crossing over, the synapsis will be maximum. See, the synapsis is maximum means the synaptonemal complex is formed and the chromosomes lie very close to each other. Now what is going to happen is the exchange of genes. So, the process of reciprocal exchange of chromosome segments along the corresponding points of the homologous chromosome is called crossing over, and this this is what simply we have explained earlier. A crossover will be formed, and as a result the chromosome will get separated. So this is known as cytological crossing over. The chromosome will undergo crossover. That is happening, and the points where the physical exchange of chromosome takes place is called chiasmata. These terms are important. Chiasmata. The point where the crossover is happening. The points where the physical exchange of chromosome is happening is called chiasmata. The places where the physical exchange of chromosome is happening is called chiasmata. So, in the cytological crossing over, chiasmata is formed. Then, the final step is terminalization. That means the crossover should happen and the chromosome has to be separated. That is known as terminalization. So, synaptonemal complex will disappear. During terminalization, the separation of the non-homologous chromosome should take place. See, they have to be separated, and that takes place in terminalization. So, synaptonemal complex will disappear. The events which takes place in terminalization are synaptonemal complex will disappear at this diplotin stage, and this takes place during the diplotin stage. Diplotin stage of meiosis. This takes place in packaging stage. So, diplotin stage, this is happening. So, they remain associated at the chiasmata, and the chiasmata represent the sites at which the genetic recombination has takes place. And as the chiasmata formation will be advancing, and that will be displaced, or chiasmata will be displaced towards the terminal ends of the chromosome, and this process is called terminalization. And the non-sister chromatids repel each other beginning from the centromere towards the terminal ends. It's actually they move apart like that of a zip. When you open a zip, it will move apart. When it is in the closed condition, it is in the it is joined together. So when you open a zip, what happens? The two sides of the zip will get separated. So likewise, it will be getting separated, and that's known as the chiasmata will move, and the two chromosomes or the non-sister chromatids will get separated. That's known as terminalization. So the non-sister chromatids will separate each other from the centromere towards the terminal ends like a zip. Then the disassembly of the synaptonemal complex takes place. So these are the three major steps in meiotic crossing over. 1. Synapsis. 2. Duplication of chromosome. 3. Crossing over, and 4. Terminalization. What is happening in synapsis? Synapsis is happening during the zygotane stage, and during synapsis the chromosomes or the homologous chromosomes will lie parallel to each other or very close to each other, and they form a structure called bivalent, and a synaptonemal complex is formed between these non-homologous chromosomes. Second step, duplication of chromosome. That happens in the packeting stage of meiosis. And during this duplication of chromosome, the homologous chromosome will get duplicated and they form a tetrad. Then 3, or the third step, the crossing over, that happens during the later stage of packeting itself. Crossing over happens in packeting itself. The exchange of the chromosomal segments happens between the two non-sister chromatids, and here a chiasmata is formed. When crossing over happens, the structure which is formed is called the chiasmata. And the final step is known as the terminalization, where the separation of the crossing homologous chromosomes will have non-homologous chromosome happens, and this takes place during the diplotene stage of meiosis. And at this stage, what happens is the chromosome will move apart from the centromere towards the tip of the chromosome. These are the major steps in meiotic crossing over. Now let's see another type of crossing over. We have learned one type of crossing over, that is a crossing over which is taking place in the gametic cells, is called gametic crossing over or meiotic crossing over, and the crossing over that is taking place in the somatic cells is called somatic crossing over or mitotic crossing over. And among those two, the one which is significant is the meiotic crossing over or the gametic crossing over. Mitotic crossing over is very rare. Now, based on the number of chiasmata formed, the crossing over is of different types. That is, single crossing over, double crossing over, and multiple crossing over. Based on the number of chiasmata formed, crossing over is of 3 types. Single crossing over means 1 chiasmata. Double crossing over means 2 chiasmata. Then multiple crossing over means more than 2 chiasmata will be formed. Let's see one by one. First is single crossing over. Single crossing over. If the chiasma formation is only at a single point of the 2 homologous chromosomes, that is called a single crossing over. Single crossing over means, for example, if you have 2 chromosomes like this, single crossover, 2 chromosomes are aligning like this. 2 chromosomes are aligning like this. Then in single crossover, let's see what's going to happen. In single crossing over, what is happening is chiasmata formed will be only one. Only one chiasma is formed in single crossing over. So as a result, what is happening? As a result, I said only, only 2 chromatids are affected when a crossing over is happening. So here you can see only 1 chiasma is formed. this is what is known as single crossover. Single crossover means the crossing over happens only at one point, and this is the point where the crossing over is happening. See, this is the region where crossing over is happening. This is the single point where the crossing over of the genes happens, or crossing over of the chromosomes will take place only at a single position, and this is known as single crossing over. So, if the chiasma formation happens only at a single point, if the chiasma formation is only at a single point, and this type of crossing over is called single crossing over, means only one chiasma is formed. That is single crossing over. Now, next is double crossing over. So, in single crossing over, the gametes formed by this type of chromosome is called a single crossover gametes. The gametes which are formed as a result of single crossing over is called the single crossover gametes. Next is double crossing over. Double crossing over. Double crossing over means the chiasma formed will be two. Two chiasmata is formed. Two chiasmata is formed as a result of crossing over. That is known as double crossing over. Means if these are the chromosomes, during crossing over what happens is at two regions the crossing over will take place. This is known as double crossing over. That means what? Two chiasmata will be formed. This is known as double crossing over. That's known as double crossing over. And in double crossing over, the distance between the two linked genes is relatively more. So, two chiasma will be formed simultaneously between them. And this double crossing over is of different types. Double crossing over means two chiasmata is formed, and this double crossing over is of different types, that is based on the chromosomes or strands which are involved. Based on the strands which are involved, they are two-stranded double crossing over, three-stranded double crossing over, and four-stranded double crossing over. If two chromatids are involved, it is called two-stranded crossing over. If three chromatids are involved, it is called three-stranded crossing over. If four chromatids are involved, it is called four-stranded double crossing over. It's like this. This is a two-stranded double crossing over. This is two-stranded double crossing over. Three-stranded crossing over means one will be like this. Then another will be like this. This is known as three-stranded crossing over. Three chromatids will be involved. If four chromatids are involved, it is called four-stranded crossing over. Now, next is multiple crossing over. Multiple crossing over means if more than two chiasmata are formed. More than two chiasmata are formed, it is called multiple crossing over. And this multiple crossing over is actually a rare phenomenon. And this is actually formed during the tetrad. And these are the multiple crossing over. You can see a double crossover, a triple crossover. I said that is if a single strand, this is two-stranded, then here these two chromatids are involved, and if three are involved, then four chromatids are involved. This is two chromatids, two chromatids are involved, here three chromatids are involved, here four chromatids are involved. In this picture, it clearly shows the crossing over between the chromosomes, and this is called multiple crossing over, and this is very significant in case of the during gamete formation in cells, and this helps in producing new combinations or new variations in a population. Hope it's clear. So the different kinds of crossing over are there based on the number of chiasmata formed. If one chiasmata is formed, it is called single crossing over. If two chiasmata is formed, it is called double crossing over. And if more than two chiasmata is formed, it is called multiple crossing over. Then, in double crossing over, based on the number of chromatids involved, it is of three types. One is if two chromatids are involved in the crossing over, it is called a two-stranded double crossing over. If three chromatids are involved, it is called a three-stranded crossing over. If four chromatids are involved, it is called a four-stranded crossing over. And multiple crossing over, it's very rare. Now, let's see the difference between the linkage and a crossover. Linkage we have learned. Linkage means the tendency of genes to remain very close to each other generation after generation. So as a result of linkage, what is happening? More parental types will be formed in offsprings. More number of parental phenotypes will be produced as a result of linkage. And the strength of linkage between the two genes increases if they are closely placed to each other. Then I hope you remember that is what is known as linkage. Linkage means if the chromosomes are, if this is a chromosome, linkage means the chromosome lie very close to each other. This is the chromosome. Linked genes are genes like this. They lie very close to each other. These two are called the linked genes. So they lie very close to each other. As a result, the parental varieties will be more during inheritance. Then it helps to maintain a newly improved variety, and so parental combinations will be more. So what is the advantage of crossing over linkage? As a result of crossing over, there is a shuffling or exchange of genes. This is very important. There is an exchange of genes or chromosomal parts during the crossing over, and that produces a recombination, and that produces varieties or variations in a population. So the frequency of crossing over between the two genes decreases if they are closely placed. So between the linked genes, the tendency of crossing over is very less. But just see one thing is, if this is a chromosome, if these are the two non-homologous chromosomes, and if one linked gene is here and if these two are the linked genes, if a crossing over occurs like this, of course there will be a separation of the linked genes. As a result of recombination, even the linked genes can be displaced, that can be separated into two different cells. So as a result of this recombination, variations are formed like that, because there is crossing over of this chromosomes. Now, factors affecting the crossing over. There are many factors which we have said once, that is the age, the position, the sex of the individual, the temperature, etc. are the main factors which affect the crossing over. One is the distance between the genes. The frequency of crossing over is actually directly proportional to the distance between the linked genes. Directly proportional means the frequency of crossing over is more if the distance between the genes is more. If the distance, that is the crossing over, of course, in all the pictures we have learned, if the location of genes is far apart, this part and this gene is very easy to recombine. So if the distance between the linked gene increases, the frequency of crossing over increases. That is distance between the genes. That is, the crossing over is directly proportional to the distance between the linked genes. Then, the chiasma formation at one point will prevent the chiasma formation in the vicinity. That's of course when you tie or put a knot like this. If a chiasma is formed like this, see, if a chiasma is formed like this, the tendency of formation of a chiasma at this location is very less. But another chiasma will be formed at a region which is far from the chiasma. So, the distance between the chiasmata will be more. That is known as interference. That is known as interference, and the factors which affect are the sex, the age of the female, then position of the genes, sex, temperature, nutrition, etc. And the chemicals, of course, I have said these are the chemicals which affect, and the distance of the centromere, the radiations will affect the recombination, plasma genes, genotype, the chromosomal aberrations, and the distance from the centromere also affects the